All right. Okay, we're going to go ahead and get started. Before I get started, I want to thank everybody that's come tonight, caring enough to learn more about science. It's really important. Thank you, thank you, thank you to the Opera House for allowing us to come and use this space. I missed the last one last week. I was on the West Coast. Apparently, someone disapproved of my travel. But the first one event was great. I heard that the talk last week was wonderful, and we're just so grateful to the Opera House for allowing us to come here and use this space, which goes back to our 50th anniversary celebration, because we used to have our cafe size here on a routine basis. So thank you, thank you, thank you, Kathy. I also want to thank we have a number of board members and former board members in the audience. Can you raise your hands? So many. In fact, the woman who hired me is right in front of me now, Louise Bowditch, which I will forever be grateful to Louise for hiring me. And our board chair, current board chair, Wendy Wolf, is in the back. So wonderful, wonderful people that give their time to Bigelow. And we are so grateful for it. And then we have many donors in the audience that I want to thank. In fact, I want to especially thank Wendy Drummond, who is here, who hosted our council event. And we had, hundreds of people swarming over her beautiful yard a couple of weeks ago. And it was a great event, and we are so grateful. This is such a wonderful community of people that give their time and their talent and their treasure to support a lab like Bigelow, and we couldn't do it without you. Every year, we like to give a great big thank you to H.M. Payson. They are a main financial advisor, and they sponsor Café Sci every year, and we are very grateful for that. So thank you, thank you, thank you to H.M. Payson. And now it is my pleasure to introduce our speaker for tonight, Ramunas Stepanauskas. I love doing these introductions because I always go back and I look at their CV and I learn something new. So Ramunas is from Lithuania. He was trained in Sweden, and he has an impressive number of languages that he speaks. Lithuanian, obviously, English, as you will hear, as well as Swedish, Russian, and Spanish. So if anyone, we could test him later in the questions. And actually, Louise Bowditch could test him later in the questions, but they're friends, so she probably won't. But Ramunas is a real visionary. I mean, we have a lot of very impressive scientists at the lab, and I think Ramunas stands out even amongst that crowd. You're going to hear about the Single Cell Genomics Center that he started at the lab along with Mike Siraki. And he has just continually pushed the envelope on helping us understand bacteria, microbes across the board in the ocean, one cell at a time, which is a little mind-boggling for somebody with the kind of career I've had where I just lumped everything together and tried to look at elements, that we can actually look at single cells. It's just incredible. So without further ado, I want to introduce Dr. Ramunas Stepanauskas.
Thank you so much, Debbie, and thanks everybody for coming here to talk to listen. Talk about microbes in the ocean, which is not necessarily the most kind of compelling topic, but I hope it's gonna. I hope to change your minds. Now, can you hear me? The end of the back rows, is that all good? Awesome. Great. So I'll talk about the research performed by my group at Bigelow, and what we do is we develop innovative molecular tools to do research on microscopic life in the ocean, and we use these tools to understand the global processes at the resolution of individual cells. And I know that most of you by now feel like, what the hell is this guy talking about? And I'm done. But hopefully by the end of this evening, there will be. I'll make some sense. And if I don't, it's on me. We scientists try to invent too many words, and sometimes it's very useful to actually cut through that and be more reasonable in our explanation of science. So when Bigelow was founded back in 1974, I was growing up in Lithuania, and Uh, and I was growing up 200 miles away from the closest saltwater body, which was the Baltic Sea. The country was occupied by the Soviets, which meant that you couldn't travel anywhere abroad. So the chances of me ending up being a scientist at Bigelow lab way less than 1 in a billion. And yet, well, the world changed since then, mostly for the better. And I went to college in Lithuania first, and then continued my studies in Sweden. Picked up some of the Swedish culture, and ended up with a PhD in freshwater ecology or limnology. Then I was invited to continue to do my postdoctoral research at the University of Georgia. And that's really where I was, for the first time, exposed to oceanography and to molecular microbial ecology. That field of molecular analysis of life and microbes included was just emerging, and there was a huge it was obvious that it potentially is huge, but at the same time, I was quite frustrated by how little you could do with these tools of that day, and there were lots of napkin drawings and discussions with my friends, both in the lab and in coffee shops, about how to improve these tools and how to make them useful to answer real big questions. And so by thinking about it and searching for a place where to try to pursue these ideas, I ended up at Bigelow. Especially exciting was the prospect of collaborating with Mike Siraki and Nicole Poulton, who were also thinking about it, and they had some unique instrumentation that was attractive and useful in that respect. And so now, 19 years later, is really unbelievable that it's been 19 years. And just this spring, our son graduated from Lincoln Academy, and to embarrass him even more, he's right here. He's heading out to college this fall. So now let's get to the marine microbe. So for most people in the world, if you would say that you study microbes in the ocean, they rightfully so think that this is really weird. Why would you do that? Why don't you do something more important? For most scientists at Bigelow, including myself, we actually went to this field just because we know how important microbes in the ocean are to the globe and to humans. And that's and so if you think about life on our planet, what is easy to comprehend for humans is the large objects that are of similar size. And on land, life is dominated by plants and animals. And if we go into the ocean, we have a totally different environment where most of life is microscopic. It's composed of critters that fit inside the tiny drop of seawater. And in that single drop, we have an entire ecosystem. And this whole world was completely unknown until we invented microscopes and other instruments to be able to detect them and to study them. And so if you look at that drop of seawater under a microscope, if the computers work, we end up seeing something like this. So here, this one big thing, which looks like a flying saucer, is a diatom. It's an algal cell in the sample. The green blobs are bacteria, and then lots of tiny little dots are individual viral particles. They are tiny individually, but their numbers are literally astronomical, and that's why they are such. At the end of the day, they are real, true shapers of the environment on our planet. So if you would take all the life in the ocean and kind of separate into different bins and weigh them against each other, we realize that carbon that's tied in marine microbes is about 10 billion metric tons. Phytoplankton adds another billion tons. And then when you get into fish and whales, they really are negligible in terms of the total carbon content in the ocean and in terms of their contribution to the biogeochemical process. And so, of course, for humans, we do have good reasons to care about fish and whales, and we do. But at the end of the day, if all the whales in the world would disappear, suddenly they would just be gone. Somebody in Kansas would not notice unless they read news. If marine microbes would disappear, the planet would become uninhabitable in a fairly short order. So if we would step aside from our human-focused view of the planet and think about the predominant life forms, for those predominant life forms, the world looks like this. Any meaningful interactions in the world actually just with your closest microscopic neighbors. Now, why humans care about marine microbes? So there are several reasons, and it's just kind of sum them up. Especially important currently is their contribution to the global carbon budget. And so marine microbes are responsible for about a third of all the carbon dioxide that is consumed, that's absorbed by the oceans. So photosynthetic microbes use it up for building their biomass. At the same time, almost all of that carbon is released back to the atmosphere by the same microbes, in addition to phytoplankton, also bacteria. And so that fine balance between the photosynthesis that consumes carbon dioxide and the respiration or breathing that takes most of that biomass and turns it back into the carbon dioxide has a huge impact on the composition of the atmosphere and the greenhouse effects of that atmosphere. Another important activity of the microbes is the production of all kinds of chemicals. So there are so many that they literally make up the composition of the seawater. They produce anything from sugars to oils to proteins to toxins and so on and so forth. And so you can think of the seawater as a soup that is being cooked by microbes for billions of years. And the composition of the incredible diversity of these microscopic living forms in the ocean is we are just starting to realize the extent of it and the impact of that diversity to the functioning of the planet's biosphere. So biologists often organize the information about biological diversity in the context of genealogy. And so just like we think about the family trees and who's related to whom, and how far back, the same way biologists organize their knowledge about the biological diversity. And so back in the days when Bigelow was founded, this was the textbook understanding of the diversity of life on planet Earth. And so the way these three major branches, which were plants, fungi, and animals, the large organisms that humans could see and easily apprehend. And then you have this kind of amorphous trunk below them that are all the microscopic forms of life. Since then, the worldview, the view of the diversity changed completely. This is how we think of it right now. And now all these large forms of life ended up in forming just a few small twigs in one side of this huge tree of life. Where the rest of it is all single-celled microorganisms. And this tremendous radical change of the view of biological diversity was primarily due to the new tools that are driven by DNA sequencing, and especially the sequencing of entire genomes. And so genome is a complete set of all the genes that an organism has. Which brings us back to year 2000, which was a very important year for biology. Some of you may remember all the headlines that we made by by the completion of the first human genome sequencing project. It was an extraordinary accomplishment. It took over a decade of work by thousands of scientists, and it cost over $5 billion to complete. But when it was done, it provided a foundation for a whole slew of new research fields, and these research fields now helping us to diagnose and to treat an increasing number of human diseases. And microbiologists, including marine microbiologists, figured that this is really powerful, and many of these same tools or their modifications can be very useful in figuring out what microscopic life in the oceans is and what they're doing. And you have to think about genomes as blueprints of life. So just like a blueprint of a car, it's very informative about how that car is built and what kind of functions you can expect from it. Much more informative than just looking at a picture. Same with microbes. If you look at that image, you see blobs, and they're kind of boring, and they're deceivingly looking like all of the same. The real information about the biological features comes from analyzing DNA and other molecular features of these cells. Now, it would be nice if we had a machine where you just take a sample of seawater, press a button, and you get all the genomes from all the species of microbes in that sample. That would be nice. Maybe in 50 years, we could expect that kind of technology. It's more complicated today, and so there are various ways how we can obtain genomes. The most traditional way is we start with a traditional microbiology where you grow up a microbe in a test tube or a petri dish. You get what we call a pure culture. So it's just a one type of microbe that grew up into many cells, and that produces enough material to extract DNA and then to start sequencing that DNA. The sequencing machines don't give you a genome, they just give a pile of what we call short reads of DNA. So they're stretches of a few hundreds of letters of DNA, they're random. And then you take that pile of reads and put it pass them through a computer program, which assembles them like a jigsaw puzzle into that genome. And so using this approach of cultivating a microbe and then sequencing and assembling the genome can produce very high quality data, but most microbes in nature, and including oceans, refuse to grow in captivity. They're just like birds. They're sad, not being in their natural environment. It's something that can be used only to analyze a very tiny fraction of biological diversity on the planet. So to circumvent that cultivation challenge, the most common approach today is called metagenomic sequencing and assembly. And I'm trying to get backward, but it doesn't work. Anyway, so the metagenome, the way it works, you take that environmental sample, like seawater or soil or whatever you're analyzing, and you don't even try to cultivate cells. You extract DNA directly from that sample and start sequencing, producing these short reads, and then you use more advanced computational tools to piece together many jigsaw puzzles at once. And so in that way, you don't rely on cultivation. You can access a more diverse set of microbes that are difficult to cultivate. It's been very useful. It does produce novel information. Unfortunately, it does not give you all the genomes, and the genomes that you get often are contaminated and have other artifacts. So that's the reason why back in mid 2000s, I started working on an alternative technique, which is single cell genomics. And what we do here is we start by a physical separation of individual cells of microbes. So we don't try to cultivate them, we just sort them out using instruments called flow cytometers and drop them into wells of microplates. And I have an example of a plate right here is it's not a big thing, but it has 384 wells, and so each of these wells gets a single microbe. And just to keep in mind the extent of the technical challenge, so here's a little test tube with 1 milliliter of seawater, and that 1 mil has 1 million of microbes and about 10 million of viruses. And so the instruments we use are able to pick these microbes one by one and then carefully drop them into a plate. And then at that point, we can start working with them individually. Instead of trying to cultivate them, we use biochemical tools to produce many copies of DNA. So we just have enzymes that copy the DNA from that single cell over and over to produce billions and billions of copies to make enough of it to sequence. And then we go through the whole route of producing short reads, then assembling these short reads into the like jigsaw puzzles. But we are dealing with individual jigsaw puzzles. We're not mixing them up. And that leads to higher quality and to also to the ability to recover genomes that are challenging to recover in other ways because of the extreme diversity of microbes in nature. And so my group pioneered the development of this technology, and back in 2009, we established the first in the world center to perform this analysis at large scale. And we figured that it would make no sense to keep that technology just to ourselves, so we opened it up to the rest of the world and started offering services of single-cell genomics. And these dots on the map here show the locations of the customers of the center. It's over 100 organizations around the globe that include universities and research labs and companies. And I'm also proud that the center now has wonderful facilities, but what we started with was this back in 2009, and it's really great. I feel like it was a bit of like a Silicon Valley startup. In addition to this triple wide, we also had a room in a truck garage and a few other places. And so one example of research that we perform using this technology and the infrastructure is we call Global Reference Genomes Project, or GORGE. And here we've been producing sequencing very large numbers of genomes from samples collected around the globe in tropical and subtropical regions. And then the goal of that was to produce a large database of these genomes. So if you think about the Human Genome Project, where the goal was to produce the genome of the human, in the ocean we have endless number of species. So it's not a single genome. It's many genomes that you have to be able to have it as a foundation for other molecular studies. And so that's the goal here. We also have other goals. And that data can be used and is being used by many scientists for various reasons. And this figure here is busy, but the point here is just one of the illustrations, again using the genealogy, the family tree kind of analogy to illustrate how diverse are microbes in the ocean. So each branch here is not just a species; it's a whole large lineage, like a family, taxonomic family level lineage. And the pie charts at the tips of these branches indicate the chemical types of chemicals that we predict from genomes to be produced by these microbes. So it's an illustration, very simplified illustration of an extent of diversity, not just the genetic diversity of microbes, but also the diversity of their capacity to produce various chemicals that are we call secondary metabolites, so bioactive compounds, which some of them potentially could be useful as drugs, as antibiotics or anti-cancer drugs. Now, genetic diversity, how do we measure that genetic diversity in microbes? So one way to do it is by analyzing the percentage of their genetic letters that are identical between the two organisms. And so for perspective, all the humans are almost identical to each other in genetic terms. They are 99.9% the same in terms of their letters of their genome. If we compare humans to chimps, they are 96% identical. If we go to cats, it's about 90%, cows and humans about 80%. Now, if we take a drop of seawater, literally just a drop of seawater, and then look at start comparing the pairs of genomes in that drop, this is how these pairs are distributed. So most of the pairs of genomes of microbes in a drop of seawater are way over here. So what it means is that most of the microbes, for an average microbe, most of their neighbors are way more distant to them than we are to cows. So most of my research concerns oceans, but I do work also in other environments. Here's an example of collecting samples in the deepest mines in the world in South Africa. And what we study here is microbes that inhabit pockets of water that are trapped in between rocks. And they've been trapped there for millions of years, and they've been totally separated from the rest of the world, and like the plants that photosynthesize, produce organic matter. So the tools that we developed and the SCGC capacity has been used to try to figure out how is that possible? What kind of energy sources these microbes use? How do they? What kind of genetic capacity do they have to produce biomass? And also, it's been very puzzling to see that how slowly they evolve. We found the same microbes, nearly identical microbes in deep underground in 3 continents, in Asia, in North America, and in South Africa. And it's been a big puzzle to understand how did they manage to stay so conserved and not essentially stop evolving for many millions of years. Another interesting project was a collaboration with the NASA Jet Propulsion Lab, where I was involved in trying to come up with better ways to detect microbial contamination on spacecraft, including this Mars rover Perseverance, which is now collecting samples on Mars surface as we speak. And so it's really important for NASA and for other space missions to ensure what's so-called planetary protection, to make sure that we are not bringing microbes from Earth to these other planets and contaminate them before we even have a chance to search for the indigenous life. So these kind of projects are really, really interesting, and they make you to think outside the box. But at the end of the day, many of our most interesting discoveries were made right here in Blue Bay Harbor, and collecting samples from Bigelow dock and from DMR dock. So that's the end of my first part of the talk, and I look forward to some questions. The next part of this talk is I have to kind of try to address the theme of our celebration of the 50th year of Bigelow, which is celebrating the past and solving for the future. And so trying to envision how the various research interests and technologies are going to be progressing into the future. And that brings us back to this view of the world from the microbial perspective and how it impacts the humans. And one of the obvious things that we have an imperative to work hard and to work fast on is the exchange of greenhouse gases between the ocean and the atmosphere. And so at this point, we have somewhat reasonably good understanding of the rates and the controls of the photosynthesis. So the algal part of the equation, which consume carbon dioxide, about a third of all the carbon dioxide that is exchanged with the atmosphere every year. We know much less about the other part of the equation because the almost all of that carbon dioxide that is taken up by algae is brought back into the atmosphere by marine microbes. And so the controls and mechanisms and the diversity of microbes involved are way less understood. We know even less about how these complex processes change as we acidify the ocean, as the temperature goes up, and as humans impact the oceans in other ways. And we also have a huge opportunity to utilize the potential of marine microbes in bioeconomy. And so what I mean here is the capacity of microbes to produce anything from biofuels to nutraceuticals to pharmaceuticals. And at this point, most of this capacity utilization is limited to the tiny fraction of microbes that are easily growing in the lab. That 99% of the diversity in the oceans and also in other environments that does not like to be kept in captivity have not been explored and have not been used in any extent to any substantial extent in bioeconomy. So that's a potential that all of these topics, I think they're going to advance substantially over the 50 years. There will be huge progress made. It's going to be a teamwork by many thousands of research groups, each of them contributing a part to it. And in the case of my group, one of the contributions that I see continuing from our current work is connecting the cell's genome, so that's genetic potential, with actual biological function. That's a huge challenge because most of the genes that we know exist in the ocean, millions, tens of millions of them, most of them we have no idea what they're involved in, what they do, and how they impact the oceans, and in which way they may be useful to humans. And so one of the instruments we use in this research is called flow cytometer. And Nicole Poulton talked about flow cytometers a lot a week ago. This illustration here shows how this instrument illuminates individual cells passing a stream and using lasers. And then we can measure optical properties of these cells and make conclusions about the composition, the chemical compositions, especially pigments of the cells. Now, in addition to pigments, what we can also analyze are various probes. So probes that turn cells fluorescent, make these cells emit various light wavelengths. And these probes can be used to detect microbes that are involved in various processes and even quantify that involvement. One example is we are collaborating with chemists who are producing these probes that are specifically targeting microbes that are involved in the production of complex chemicals that are called polyketides and non-ribosomal peptides, which are interesting from pharmaceutical point of view. So we're searching for new microbes that may be a source of antibiotics and anti-cancer drugs. And we are searching for them by collecting samples of DMR doc. There's a lot of interesting biology that we find. This example here illustrates one of the clusters of genes that we found to be almost identical to the cluster of genes that encodes the production of bubonic plague toxin. That's quite scary, and it's interesting. It just indicates this probably fierce chemical battle that's going on DMR dock between microbes. Another probe that we've been working quite a lot on, it takes us into a totally different direction. So this probe allows us to measure the rate of respiration or breathing of individual microbial cells. And so by doing this analysis and also sequencing genomes and using analyzing other type properties of the cells, we realized that the most abundant microbes in the samples we worked with hardly breathe at all. They really hardly consume any oxygen and release any carbon dioxide. And the way they probably can do that is by, instead of breathing to produce energy, they complement the energy sources by harvesting sunlight. And it's not photosynthesis, it's totally different system. It's called rhodopsins, so light harvesting using rhodopsins. So by using that system, they gain energy and don't rely on respiration or breathing to the extent that we usually think of them to be dependent on. So all this work was done in Boothbay Harbor and was recently published in Nature. And what we are hoping is to expand that geographically and get a much better sense of how important this process is on a global scale and to what extent that process impacts the balance of carbon dioxide uptake and release from the oceans. Another cutting edge technology that we're working on is focused on viruses and other tiny extracellular genetic elements. And so what we do, and these particles are even harder to study than microbes. They're super small. They're so small that that instrument that I talked about, flow cytometer, is not sensitive enough to analyze. And so what we are trying to develop instead is a totally different approach where we are encapsulating tiny droplets of seawater inside shells of gelatinous material. And then we are able to manipulate these millions and millions of these capsules in a single tube like this simultaneously to analyze thousands and millions of individual viral particles. And the additional fun factor of this project is that we are collaborating, pushing this technology forward together with a startup company from my old home country, Lithuania. This is this figure illustrates some pilot results that we just processing right now. It's not published yet, and each of these dots represent a genome of a single particle. Some of these particles are cells, some of them are viruses, some are something else that we are still figuring out what they are. And so each dot represents this entire data set of DNA sequences and genes that these sequences encode. So the importance of this technology is, one is that we are now capable of analyzing entities in the ocean and in other environments that were not possible to analyze before because they are so small. The other benefit is that it's much cheaper. We're cutting the cost by about tenfold, and we can produce about tenfold more data. So that kind of has a promise of democratizing the whole field of microbial single-cell genomics. And there's a potential of using these tools also in the context of human health. So we study microbes and viruses in the ocean, but potentially that same technology could be helpful in tracking, for example, new variants of viruses that infect humans. So for that purpose, we have a patent application pending to apply these tools in that kind of human health context. And the other crazy thing about those results that I just showed is that they come from super tiny droplets. So they come from 250 nanoliters of seawater, which is about 1,000th of what is in this test tube. So it would be even invisible with a naked human eye. And from that one single tiny droplet, we are getting over 2,000 genomes of viruses and cells. And I think that's really, it makes me really wonder about nature. It's incredible how nature works, and it's also it's an example of how by analyzing analyzing nature at the most fundamental level, we are creating the type of knowledge that's needed to understand and for the stewardship of our oceans at the global scales. So I want to finish with thanks to my group, current and many former members, and I'm sorry I didn't make the list complete, as well as the funding agencies. And thank you for getting through the talk about marine microbes, and I look forward to a few additional questions.